statements contained in this presentation have not been evaluated by the Food and Drug Administration. The products discussed are not intended to diagnose, treat, cure, or prevent any disease.
Okay, today we'll be having Dr. Oclarit, who will be discussing on genetic engineering and its medical applications. Okay, Dr. Oclarit, please take the floor.
Good morning, everyone in the Philippines, and good evening in United States. Today's topic is a little bit technical, and I don't know if it's all up to you to get some correlations to our product, but our discussion today will be mainly academic in nature and how genetic engineering is being exploited in medical therapeutics and intervention nowadays. So the genetic engineering is the wave of the future for a lot of things in science, not only in medicine, but also in agriculture, fisheries, and pharmacology. So the objectives of this lecture is for you to learn the basic tools of genetic engineering, how gene cloning is carried out, how the DNA is used to identify living organisms in DNA fingerprinting, how to detect very tiny quantities of DNA even when they are millions of years old, just like in Jurassic. Have you seen the movie on Jurassic? That is millions of years, and yet the DNA was detected and was magnified and later on developed. In addition, ethical problems of this genetic engineering will be presented and some possible ways of dealing with them considered. So descriptions of molecular biology. Molecular biology is the area of biology that is concerned with how biological molecules interact with each other at the molecular level. Molecular biologists use variety of sensitive procedures or very intricate procedures to study the molecular interactions. And molecular biology describes techniques that are currently available that allow us to control genetic chains anywhere in the genetic code of a DNA of a particular organism, including human being. So we can now control the expression through the advent of molecular biology. We can now control the expression of the genes in the DNA of all our cells. That's why our product stands as a molecular manipulator or a molecular intervention against the background of our cellular interactions and functions. The history of genetic engineering started in the 1950s and 1960s. It was only in the 1970s when the DNA was isolated and Dr. Hamilton Smith accidentally found that a certain enzyme in bacteria, we call it DNase. This is the bacterium Haemophilus influenza, which is actually the cause for flu. They have found out that this Haemophilus influenza was able to release an enzyme that enabled to cut DNA only at a unique site or a unique DNA sequence. Remember that DNA are just letters of A, T, G, C. A pair with T, G pair with C. And our DNA is a sequence of letters, no, A, T, G, C. And this enzyme DNase coming from bacteria can cut certain segment of DNA in order to transfer. So if the tailors and dressmakers have scissors, molecular biologists are provided with DNase. These are the enzyme that can cut the DNA at unique sequence. And that unique sequence of the DNA is known as the palindromic site. So this particular DNA enzyme that cut DNA at particular sequence in order to isolate or transfer the DNA to another site, and that cutting site there is known as the palindromic site. Do you know the meaning of a palindrome? Okay, The term palindrome are set of letters that can be read the same forward and backward. For example, in this illustration, you examine very well the sentence, Dennis seen can also be read Dennis seen backward. Then there is another palindrome, sex at noon taxes. So you read it SEX from backward, SEX, sex at noon, taxes, also the same. Another palindrome is Madam, I'm Adam. So that's another palindrome. Ma is Anan, as I am. Did you get the idea of palindrome? Palindrome are set of letters that can be read the same forward and backward. Okay, you have those example and it will ring on your mind because these are interesting sentence. This enzyme called DNase can read and cut the DNA at the palindromic site. So palindromic site, remember that. So a palindrome is a site or a sequence of base pairs in a double-stranded DNA that trades the same backwards and forward across the double strand of the DNA. Example, you have the example of the sequence of base pairs. So GAA, TTC. What is the pair of C? G. No, backward na ta, backward. What is the pair of C? G. What is the pair of T? Diba A? What is the pair of A? T. Then what is the pair of G? C. So let us now try to read it backwards. So forward, GAATTC, if you make complementary pair in the opposite side and read it backward, it is also GAATTC. So this is an example of a palindromic site in the DNA. That enzyme recognizes the sequence, palindromic sequence, which is GAATTC. And it was found out that will cut between the G and A. And it will cut on the G and A on the opposite side of the DNA double-stranded molecule. GAATTC is actually a palindrome. That is a sequence of DNA that is recognized by the enzyme. And that enzyme comes from bacteria. And that enzyme is termed as the restriction enzymes. The restriction enzymes, therefore, are those enzymes that cut at palindromic site, and they are called restriction. That's why they are the term restriction enzyme, because these are the enzymes that are specific, that are restricted to that site only. So the enzyme recognizes that specific sequence. That is why they are called restriction enzymes. Based on the types of the cuts, they can either make two types of cuts. These are what we call sticky cuts and blunt-ended cuts. Let's try to see the sticky cuts. The sticky-ended cut, one group cuts the strand of DNA off the center of the palindrome site, but between the same two bases on the opposite strands. So there is an example, GAATTC. The restriction site is cut between G and A of one side and another G and A of the opposite side. So making it what we call sticky ends. Why is the term sticky? Because naturally, let alone in a solution, G will always look for C and A will always look for T. That's why they are naturally paired. Therefore, without any manipulation, if you have a DNA with a sticky ends like that, they will naturally combine without difficulty because they are called sticky ends. So if you cut one source of DNA by that restriction enzyme and you cut another DNA by the same restriction enzyme, you are now assured that those two DNA of different sources will now anneal or combine because of their sticky ends. The sticky ends are naturally paired. So you call it that sticky ends. So this leaves one group, one basis overhanging on its opposite strand. Such ends are called sticky ends because they form the hydrogen bonds between the pairing of nucleotides. Remember that nucleotide pairs are composed of hydrogen bonds. And these hydrogen bonds are natural bonds that makes the DNA sticky to each other, because given a natural solution, if you put guanine in water, it will look for cytosine. If we put thymine in the water, it will look for adenine. That is why they are naturally sticky to each other. That's why they are called sticky ends. So if I may ask, what makes the DNA sticky to each other? It's because of the hydrogen bonds. So if there are sticky ends, there are also blunt-ended cuts. Another type of cut is a straight across the double strand, is known as the blunt-ended cut. Blunt-ended cut, cutting restriction enzymes cleave the DNA between two bases in the middle of the palindrome. Note that there are no overhangs. The cut ends of this DNA is compared with the previous figure. So let us try to look at the sequence where it is cut. TGG, CCA. The palindrome TGG, CCA is cut between G and C of one strand and G and C on the other strand. So if you look at the upper, it is cut already at G and C, and then at the other ended is also G and C. So therefore, the resulting DNA that is cut is known as blunt-ended cuts. So, and then once the DNA is cut, the DNA can be combined again using the enzyme called DNA ligase. So after the DNA is cut, it will be combined again by the enzyme called DNA ligase. So ligase is an enzyme that covalently joins the sugar phosphate backbone of the DNA, because the sugar phosphate is the backbone molecule of the DNA. It is cut there at sugar phosphate backbone, so it will be joined still at the sugar phosphate backbone using the enzyme called ligase. The ligase reverses the action of the restriction enzyme, cuts the DNA. The DNA ligase will connect the DNA again. So you look at the GAA-TTC there. So the GAA-TTC will be connected again covalently by the DNA ligase. So let us now try to review the components of genetic engineering. First of all, you need a double-stranded DNA containing palindromic restriction enzymes or restriction sites. So you cannot cut a DNA until you know the palindromic site. Many types of restriction enzymes cut or cleave the palindromic site in the double-stranded DNA sample, forming either a sticky or blunt-ended cuts. The enzyme ligase will now combine the cut DNA at the phosphodiester band, or we call phosphate sugar backbone of the DNA molecule. I repeat, the backbone of the DNA strand is the phosphate sugar backbone. And that is where the DNA will be cut. And that is also where the DNA ligase will combine again to form phosphate sugar backbone. So this is a caricature of the ligation process. So for example, the DNA carrier plasmid, and then the one in yellow is the DNA that you're interested of to join. Then since they have sticky ends, so you will notice that the sticky ended there is GATC. And then on the other side is also GATC in the opposite side. So the green one is your ligase that will now connect the phosphate sugar backbone of the DNA. Therefore, what is the importance of knowing this restriction enzyme cuts? If you cut a particular DNA source of using that restriction enzyme, and you will also use the same restriction enzyme to cut the DNA of the carrier, then you will be assured that the DNA that you want to insert will connect to the DNA where you want to insert it because of the sticky ends, or you call it the restriction sites, which are palindromic. So that is how the DNA is ligated. Ligation means to bind together. The meaning of ligation is to bind again, no? To bind the fragments. So you bind one fragment to another fragment using the ligation enzyme called DNA ligase. So why is genetic engineering? The idea of genetic engineering is to move the gene of interest from the original source, say human, into another target DNA, which is bacteria, for example. So you can now get the DNA from human and insert it into bacteria. Now, why is it that you can insert it? Because of the palindromic restriction sites, which are actually the same site where the DNA is cut and where it will be joined together. So in genetic engineering, the idea is to move one gene source to another gene, or from one DNA of one source, say human or plant, then you combine that DNA from human to bacteria. So the process of moving a gene from one DNA molecule to another is known as cloning. So that is the concept of cloning. So when you clone, that means you are going to transfer one gene to another source. In genetic engineering, a fragment of DNA is cloned, moved into a vector. The vector is the carrier now of the gene, where it can be grown and copied, and it will be multiplied in large quantities, can be manipulated in many ways, and that is the object of genetic engineering. Manipulation, the word manipulation, it can either be increased or decreased, or turn on or turn off, depending on what you want to do. And this is where your genetic engineering is very important in medical therapeutics. So what are the basic steps in cloning? So I have already told you before that in cloning, what are the steps? There are 6 basic steps in cloning. The first one is to isolate the DNA of interest, isolation of the source of the DNA, and isolation of the vector or the carrier of the DNA. So in the drawing that you can see there, our carrier of the DNA is plasmid vector. The word vector is carrier. So the plasmid is actually coming from bacteria, which can now carry the gene of interest. So the foreign DNA will be cut using the restriction enzyme, and whatever restriction enzyme that you use to cut the foreign DNA will be the same restriction enzyme to cut your vector. So in the idea there, look at the foreign DNA, it is cut by restriction enzyme, and then you form the sticky ends. And since the plasmid is also cut by the same restriction enzyme, then you're assured that your foreign DNA can now be inserted or connected to your vector. You see there, the sticky ends of the vector and the sticky ends of the source of DNA are now the same. So they can now pair together and can now be inserted. Once the DNA is inserted into the vector, you call it gene. And this clone gene can now be reinserted back to the bearer of, to the cell where the plasmid is found. And you can expect the plasmid to multiply by itself inside the host bacterial cell. Since the plasmid is an extra chromosomal unit of the bacteria, then the plasmid will now be reintroduced to the bacteria. And once the clone gene will now be inserted into the plasmid, as the bacteria will multiply, your clone gene can also be multiplied. And that is the process of inserting a DNA into the vector. For example, insulin gene is inserted into the plasmid. The plasmid is restored back to the bacterial cell, and the bacterial cell will be grown in bacterial medium. They can now grow and multiply. And as the bacteria will grow and multiply, the gene that you have inserted will also grow and multiply. What are the uses of the clone DNA? Number one, the number one purpose of cloning is to multiply the gene of interest. So if you have only one copy of the gene, you insert that copy to the plasmid, you insert the plasmid to the host, the plasmid will be multiplied, and as the plasmid is multiplied, your gene is also multiplied. So one purpose of plasmid amplification, one purpose of cloning is to multiply the DNA into several copies. And another is after you have multiplied the gene, you can now use that gene for sequencing. So because you cannot sequence the DNA if you have only one copy, you have to have thousands of copies of the gene in order for you to sequence it. And then another purpose of cloning is for mutation. So you clone a gene in order to mutate a particular gene and find out what is the function of that particular gene. So nowadays, that's why in medicine, they have already discovered a lot of functions of the genes by simply mutating them in culture. And another purpose of cloning is to create humongous quantities or large quantities of the gene product. For example, in the previous treatment of diabetes, they will harvest all the pancreas of all the animals like carabao or cow. And then they will extract the insulin from the pancreas of the cow or from the pancreas of a carabao or the pancreas of a pig. And then they will purify it and sell it as a medicine for diabetes. So insulin for the cure of diabetes. Nowadays, you don't need to kill an animal or harvest the pancreas of the carabao, because what can be done now is you get the gene of the insulin from human, you insert it into the plasmid, and you restore the plasmid to the bacteria, and you grow the bacteria, and you will have the product. The product is insulin. So nowadays, the one that is sold in the market is no longer the insulin coming from animals, but human insulin produced by bacteria through genetic engineering. The insulin gene can now be inserted into the bacteria and let the bacteria produce the product. So you can create a lot of products using a clone DNA. So this is one important purpose of genetic engineering, to obtain a product that is difficult to obtain. So before, it is very difficult to obtain insulin. Nowadays, there is no more difficulty because there is already a cloned human insulin gene that is inserted into bacteria, and now the bacteria will produce the human insulin. So truly human insulin, but produced by a bacteria. That is now the importance of genetic engineering. So you can create a large amount of transgenic organisms. So transgenic organisms, transgenic means across species. So transgenic organism is an organism that contains the DNA across species. For example, bacteria, you insert a human gene into the bacteria, then the bacteria is already considered transgenic. So for example, you have a bacteria that is modified, then that modified bacteria is called transgenic bacteria. There are so many species of organisms that have already been transgenic nowadays. You have the potato, BT potato, BT papaya. The BT there is the gene from the bacteria. BT means Bacillus thuringiensis. It's a kind of bacteria that is found in the soil that can be transferred to any of the plant. And the good thing about this bacteria is that it will resist rotting. So if you insert a gene from BT, then you insert the BT gene to potato, then the potato will no longer rot. If you insert the BT gene to tomatoes, then that tomato will not rot. You will notice in the grocery store nowadays, the tomato is already very, very, very ripe, but it is not rotten because it has a BT gene. So that is a transgenic. So many of us are already beneficial of this transgenic organism. You have the BT soybeans, the BT corn, BT papaya, BT tomato, and there are so many transgenic organisms already containing the gene of interest that is transformed into the gene inserted into the plant, the gene inserted into bacteria, the gene inserted to human, and that is what we call transgenic organisms. Molecular biologists to manipulate the DNA at different organisms. These procedures used to produce better crops. So there are no better crops, as I told you. The crops that are able to resist the rotting, because rotting is due to fungus. And so this gene can resist the rotting. Human insulin can now be produced using bacteria. So however, the same techniques can also be used to make a powerful pathogen. That is why biological weapons nowadays are obtained using genetic engineering. Biological weapon is obtained through genetic engineering. So genetic engineering or cloning is used in DNA fingerprinting. It used to be we are identified through our fingerprint, but nowadays you can now be identified through your DNA fingerprint. How is DNA fingerprint done? There are three basic principles required to understand the fingerprinting. First of all, the ATGC pairing, no? A pair with T, G pair with C. And specificity of the enzyme activity, particularly the restriction enzymes. So if you get your DNA and you use a particular restriction enzyme, you can make several fragments of your DNA, and then you do a separation of those fragments, and lo and behold, you can have now a fingerprint of your own body. So mutations can remove also or eliminate the restriction in site from a gene. So by using the restriction enzyme, you can cause mutation. And what is the importance of mutation in genetic engineering as well as in medicine? By mutating the gene at a particular site, then you will be able to identify the function of that gene by means of mutation. And how do you mutate it? You mutate it by using a restriction enzyme. You cut it, you transfer it, and then you observe what are the results of those transfer or mutation. So genetic engineering is now used for diagnosis. It is now used for treating diseases, cutting the DNA at specific site. If the DNA is damaged or defective, then that damaged DNA can be cut using a restriction enzyme, and then you can insert a new DNA into the site, and then you can now be repaired. And lo and behold, the genetic disease can be eliminated using a DNA manipulation. So here is an example of a DNA fingerprinting. So here is an example there. You have gene 1 and gene 2. The red line represents the restriction enzyme cutting restriction site for enzyme 1. Then the green one is the restriction site for the restriction enzyme 2. So look at gene 1 and gene 2. The red restriction enzyme and the green restriction enzyme. So those lines there that you can see are the cutting site. So if you are going to look at the red restriction enzyme, how many cuts? 1, 2, 3, 4, 5, 6, 7. So there are, for gene 1, the red restriction enzyme has how many cutting sites? 1, 2, 3, 4 for gene 1. And then on gene 2, they have 1, 2, 3. There are 3 cutting sites in gene 2. For the green restriction enzyme, for gene 1, there are 2 cutting sites. For gene 2, there are 1, 2, there are also 3. So as you can notice here, that if you are going to do electrophoresis and separate the fragments, in the gene 1, you have how many fragments? You have 1, 2, 3. So how many fragments of yellow gene? Since red restriction enzyme will cut how many? So these are, so the fragments there represents now the cut DNA using the restriction enzyme. So if you use one restriction enzyme, for the red restriction enzyme, you have how many fragments? 1, 2, 3, 4, 5, 6, 7, 8 fragments. So you have in your fingerprint, there are 8 bands. 1, 2, 3, 4, 5, 6, 7, 8 bands. For the green restriction enzyme, you have how many fragments? 1, 2, 3, 4, 5, 6 bands. So using a particular restriction enzyme, you have a particular DNA fingerprint. And then using another restriction enzyme, it will now change the picture of your fingerprint. So in this figure, the genes with the palindrome sites for 2 different restriction enzymes, the red and the green, the number of the size of the DNA fragments that would result if these two genes were cut are shown there. So for restriction enzyme green, there are 8 fragments. For restriction enzyme red, there are 6 fragments. 1, 2, 3, 4, 5, 6. So each group of restriction enzyme produce different fragments would now represent unique fingerprint of that DNA. So if you ask me, Doc, supposing you are going to use one restriction enzyme, then consistent naka-restriction enzyme so that you will have a consistent fragment of your DNA. And the fragment of your DNA will now be separated by the process known as electrophoresis so that you can now segregate the different fragments of your DNA, and each fragment represent one band, and therefore all those bands represents what you call the DNA fingerprint. So this is the gel electrophoresis, no? It is a kind of procedure where the fragments of DNA are migrated depending on the size. So since the DNA is a negatively charged molecules, then in electrophoresis, you have a positive and negative terminal. So you introduce the gene in one side, and later on, if you run electricity, since the gene are negatively charged, it will move towards the positive side. They are separated. So DNA fingerprinting works because of mutations. The variations in genes between individuals decreases the likelihood that the restriction fragment pattern of 2 individuals are the same, unless they are individual twins or identical twins. The variation in gel fragments from different members of the same species is called restriction fragment length polymorphism, which is used in DNA fingerprinting. So this is a typical illustration of the application of DNA or genetic engineering. Do you know the case of O.J. Simpson, the pick of the crime? There is an unknown cell in the scene. It was isolated, and the O.J. Simpson cell was also isolated, and later on was fragmented using the restriction enzyme, and then the unknown DNA in the red one reveals that there are common fragments compared to O.J. Simpson's DNA, and therefore O.J. Simpson was the murderer in the crime scene. And you will notice in the green one, if you compare the DNA fragments of the red, which is the unknown, and the DNA fragments of O.J. Simpson, which is actually the green, you will notice that there are common DNA fragments, meaning to say that the culprit of the crime is identified using genetic engineering. So hybridization is actually the principle behind the identification of the DNA fragments. The principles in hybridization steps are the DNA strands are separated by breaking the hydrogen bonds, and then it produces a single strand, and then attach it to a cellulose paper, and then you are going to use what you call probe, and then once the probe is there, and then supposing the probe will now pair complementarily with the data that it complement to the DNA in question, and therefore it will later on be identified whether the gene is present or absent using hybridization. The principle behind hybridization is the pairing of GC and AT. So hybridization probe. So the purpose of the probe is for you to identify whether the fragment of interest is present or not. So you cut the DNA, and then you separate the DNA, and you insert the probe. And if the probe, for example, tubercle bacilli in your saliva, if you have a particular tubercle bacilli, so you obtain the DNA from your saliva, and then you cut the DNA, and then you fragmentize it, and then you make a probe, and the probe is actually isotope identified. Therefore, the probe will report whether it is present or absent. Because if the DNA will now hybridize because of pairing, then using the probe, you can now identify whether the DNA of interest that you want to find out is present or not. So for example, I'll give you, you extract the DNA from your mouth. So there are bacteria there. So then you make a probe. What kind of probe are you going to use? you use a probe for tubercle bacilli. So the DNA of the tubercle bacilli as your probe. And then you make the fragmentized DNA of yours from your mouth, and then you now introduce the probe. And since the probe is complementary with the DNA that you want to search, for example, tubercle bacilli, they are now going to bind together. And now you can identify whether the DNA is present or not. So that is the application of the genetic engineering, or in identification of the unknown DNA you want to find out. If I want to find out whether you have asthma gene or not, I will use an asthma probe. So I will obtain first the DNA from your blood, then fragmentize your DNA, and then attach those fragments to a particular cellulose paper, and then introduce the probe, and then hybridization na. Then, for example, I will use the asthma gene. I will now try to find out whether asthma gene is present in you or not. If it is present, then it will be identified by fluorescing using the isotope. So it's now easy for us to identify whether your gene is defective or not using a probe, because the probe is a reporter and it will report whether the gene that you want to find out is present or not. So this is an example of the DNA fragment with a probe. The probe is labeled, and therefore once they are labeled there and they are going to bind, then you will be able to identify the fragment. So the hybridization procedure will allow you to, this is now the probe, the probe will now report whether the gene is present or not. So labeled probe, a portion of the DNA, then you have there the, have you seen about the Bill Clinton case about si Monica Lewinsky? The Monica Lewinsky is the owner of the blue dress. The Bill Clinton, sperm cell ni Bill Clinton was found out in the blue dress. So the DNA in the blue dress has revealed that since the DNA in the sperm cell of Bill Clinton was found in the blue dress of Monica Lewinsky, then it was positive that Bill Clinton had sex with Monica Lewinsky. That's why using the DNA probe. So now you can identify whether you are the person, the murderer, or the culprit in the crime using a genetic engineering technique. So in this figure, 3 different probes were used, A, B, C, and are hybridized against the separated membrane-bound DNA fragment shown in the right. The 3 duplicate gels as an exercise determine which bands each of the 3 probes will hybridize with the light up. So you will notice that the blue dress of Monica Lewinsky and Bill Clinton have matched fragment, and the red one. So this is the example of the gel, the fragments of the DNA that is isolated, gel electrophoresis ang tawag. So you see the fragments, those are the DNA fingerprint. So the fragment that you see there are the bands of the DNA of different sizes. The bigger the DNA, it's in the upper portion. The shorter the DNA is in the lower portion. So you have a DNA fingerprint. So this is the example of the extraction of the blue dress of Monica Lewinsky, and the DNA was extracted there, the sperm cell of Bill Clinton. It was collected and mixed with PCR, and then it was later on hybridized, and then it was then revealed that there are common bands of DNA between Monica Lewinsky's DNA extract and that of the DNA of Bill Clinton. So in polymerase chain reaction, you have a target DNA to be copied, then you have about single-strand primers, and then you have the free nucleotides that is available, and then you have the DNA polymerase to synthesize the DNA, and of course the buffer and magnesium ions. So here is the polymerase chain reaction, or the PCR works. 1 copy of the DNA can now be multiplied in a logarithmic cycle. The first cycle, 1 copy, the second cycle, the third, the fourth, and then so on and so forth until all the DNA is copied in numerous amount. So in summary of the DNA fingerprinting, the DNA fingerprinting is a powerful technique that is capable to distinguish every individual from each other in this planet. So your DNA fingerprint is different from mine. Even identical twins have some differences. The major limitations of DNA fingerprinting are human error and the quality of the laboratory that is used. So the discovery of the PCR or the polymerase chain reaction increased the sensitivity and versatility of the DNA fingerprinting. And these techniques will allow infectious diseases to be identified using DNA fingerprinting within hours or even minutes. So with the advent of genetic engineering, the identification of diseases can now be, it used to be to identify tubercle bacilli, it used to have 2 weeks. But using the PCR, you can now identify tubercle bacilli in 2 hours or even less than that. So applications of genetic engineering in fighting diseases. In genetic engineering can be used using a signal transduction gene therapy. A gene that makes a therapeutic effect could be inserted into the site where the gene is defective. A gene or genes that regulates the production of therapeutic genes can also be inserted or multiplied or expressed. So it can also be used in blocking the damage of arthritis or any other disease. So the applications of genetic engineering is in the identification of diseases and insertion of the genetic, cutting the gene of interest from one site and transfer it on another site to express or overexpress it is actually a leap in science that enable us to apply what you call gene therapy. So gene therapy nowadays, you can have a gene of interest and then you can deliver it to the site using genetic engineering and signal transduction techniques. Thank you very much, and that's all for today.